Hello my students, I'm Madam Joyce from Educare TZ. I'm teaching biology and today I'll be with you, my lovely student, to discuss our topic which is found in Form 5, and this is cytology, whereby in our topic we are going to deal with our subtopic which is the types of cell. Whereby in the types of cell now we are discussing on the structures which are found within the cell, that is called the plasma membrane or the cell membrane. Within the cell membrane, it came up with different argument of scientists, and we have seen the first, that is the Daniel-Davidson model, and now that model we have seen also the weaknesses. So as a result now there is another model that came up that correct the Daniel-Davidson model, and that model it is called the fluid mosaic model. In previous session we tried to see this fluid mosaic model and how did they correct the Daniel-Davidson model. So now my students, let's see what is this fluid mosaic model in general and the strengths and weaknesses of this fluid mosaic model. So, fluid mosaic model is this just like observations of this fluid mosaic model. So, lipids and lipid soluble substances allowed to cross the hydrophobic region, while non-lipids are not allowed to cross. So remember within the this model, that is fluid mosaic model, wakasema kuna two parts that are found in the phospholipid bilayer, hydrophobic layer pamoja na hydrophilic layer. Sasa according to their observation wakasema kwamba lipid and lipid soluble substance are allowed to cross the hydrophobic region. Remember, this is hydrophobic. Hydrophobic meaning that water hating. So it allow material material mengine mbali na maji. So material haya ambayo yanaruhusiwa kupita kwenye this region, that is hydrophobic region, are lipid and lipid soluble substances. Lakini non-lipid hapa tunaongelea protein, tunaongelea water and other carbohydrate. They are not allowed to enter through this region, that is hydrophobic region. And we can say that large molecules such as glucose, fatty acid, glycerol, and amino acid are repelled by the hydrophobic region, and therefore they diffuse through special transport proteins called the channel proteins and carrier proteins by facilitated diffusion. So zile materials zingine zinakuwa zinaingia ndani ya cell kwa kupitia sehemu gani? Hapa ndipo ambapo sasa tunasema kwamba yale material mengine, kwa mfano tuna glucose, we have fatty acid, glycerol, amino acid, they are repelled in the hydrophobic region. Kwa sababu gani yana repel? Kwa sababu hii hydrophobic region does not allow any kind of material except the lipid and lipid soluble substance only. And here we have seen that this material, for example, glucose, fatty acid, glycerol, amino acid, and so on, they tend to enter within a cell through a special transport protein. In the diagram of the fluid mosaic model, we have seen those channels that allow different material to enter. So what are those material? These material, they tend to enter through those special transport protein, and kama yale material hayataweza kuingia through this region is whereby now we have carrier protein and facilitated diffusion. So material, they will tend to diffuse to enter through the plant or to enter through the cell. And also we can say now let's see the strengths. The strengths of the fluid mosaic model. What are the strengths? Je, hii model ina uimara gani mpaka tukaweka sasa nao tukaiamini kwamba within the cell membrane it's true now there is a different proteins, we have hydrophobic, hydrophilic, and such material that they enter and so on. So what are the strengths of this model? So the first strength of this model, they realize the presence of phospholipid bilayer and protein layer. Walikubali kwamba ni kweli within the plant cell there is phospholipid bilayer pamoja na protein layer. Remember, this become the strength within the Daniel-Davidson model. So wale wanasayansi ambao walikuja kwenye hii fluid mosaic model, tuseme labda wameendeleza ile model ambayo ilikuwa ya mwanzo ya kina Daniel-Davidson model. So this become the strength. Walikubaliana na ile model ya kwanza. And the second strength here, we can say that it also realized the presence of pores in the membrane and the presence of cholesterol, the cholesterol in the membrane. Also walikuja wakasema kwamba within the cell membrane there is a pores. Within that pores is whereby also there is a presence of cholesterol in the membrane. Cholesterol, it has a lot of function within within a cell. So become that is the strength. They recognize the presence of cholesterol and the pores within the membrane. And also the model explained the presence of polar head, that is hydrophilic, and non-polar, that is hydrophobic tail in the phospholipid. So pia hii model it become as the strength, that is within their model they tend to explain, that is this hydrophobic region and the hydrophilic region, whereby the hydrophilic region is the polar head. Polar head meaning that water loving, tend to allow water material or solvent to enter within a cell. But also they realize that or they explain the presence of non-polar head or non-polar tail, that is the hydrophobic tail, does not allow solvent material to enter rather than lipids and non-lipid material. And now we can say that the models explain the presence of glycolipid as the receptor that allows the cells to respond to a chemical messengers which regulate the activities of the cell. So now simply we can say that hii model ndio ambayo ili recognize the presence of glycolipid. In the structure of it we have already seen kwamba kuna presence of glycolipid. So ile glycolipid ina act as a receptor that allow the cell to respond to a chemical messengers which regulate the activities of the cell. Remember, while we are discussing on the Daniel-Davidson model, we were looking at the weakness of that model. Wakasema kwamba hawakuelezea kivipi cell inaweza kugundua in the outside environment there is different material that are needed to enter in. Hawakuelezea, but this model tend to explain within the cell membrane there is a presence of this glycolipid. So within this glycolipid it acts as a receptor. You know, receptor, it acts as to receive any information on the outside environment, so that that material needed within the cell activity they will enter. So that is the role of glycolipid within a cell membrane. Now also another thing, that is it is true that the membrane is dynamic, that is ever changing in structure with the phospholipid in constant motion. So hii pia waliweza kucorrect ile weakness ya Daniel-Davidson, kwa maana kwamba Daniel-Davidson walisema membrane ni static, does not change. So walivyofanya utafiti zaidi wakagundua kwamba ile membrane ni dynamic, it can change. Kwa sababu gani? It ever changing its structure with phospholipid in constant motion. So that's become their strengths. So kitu chochote kinachokuwa na strengths, obvious lazima kutakuwa na weaknesses. So every day scientists wanafanya kazi investigate new things. So now what are the weaknesses of this model? So simply we can say that the weakness of the model is that it does not indicate or explain the presence of electrolytes in the plasmalemma. So though waligundua vitu vingi sana, glycoproteins, glycolipids and so on, hawakugundua kwamba hawakuelezea kuhusu the presence of electrolytes in the plasma membrane. So within the plasma membrane also there must have electrolytes. When we are talking about electrolytes here, we mean those nitrogen ions, chloride ions, potassium ions. Those are the electrolytes, examples of electrolytes. So this model did not explain that within the plasmalemma there is a presence of these electrolytes. So my student, for more clarification on the weakness of the fluid mosaic model, I would like to invite you in the next part, whereby we're going to clarify more about the weaknesses of the fluid mosaic model. Thank you for listening.